Welcome to the presentation discussing the development of optimized beta-glucosidases. My name is El-Halabi, and my mentors were Dr. Kim as well as Emery Conley. We open this presentation with the discussion of lignocellulosic waste. On the left, we have some examples of this, which is actually plant matter that has already gone through some other sort of process and has been ready to be discarded. So as we can see, some examples are going to be apple waste as well as leftovers from corn. Now, molecularly, these are made out of cellobiose, which we see an image of on the right. This is going to be made of two carbon rings coupled together, and this differs from maltose, which is another compound that's in the same family. And so we are interested in actually breaking the bond between these two carbon rings to make something called D-glucose. So the hydrolysis from cellobiose to D-glucose would allow us to accumulate this glucose, which can then be fermented to actually make bioethanol, which is a more sustainable way to make ethanol and biofuel in general. Now, we decided to begin with an organism called Pyrococcocerosis, or PF, which is actually hyperothermophilic. So this primitive bacteria lives in a very high temperature. And so the enzymes that it makes must also exist at this extremely high temperature, which is good for more industrial processes. In essence, it has the traits that we're interested in, uh, high thermostability, but it might not have the best activity as an enzyme. On the other hand, we looked at the organism Thalormyces amistoclei, which is a fungus, or TA, as we refer to it. This fungus is not as extreme. It lives more at room temperature, but the enzyme that it makes is able to conduct the processes at a higher rate. And so we see the two enzymes in question. The PF on the left is a tetramer, so it actually has four pieces that come together to stabilize each other and to make the reaction go forwards. Now, on the right hand, we see just a monomer of TA, and it's able to conduct the reaction at a decent rate. So what we did was we put these enzymes into E. coli, which is a bacteria that we can grow in the lab. And what we would do is we would place them on a petri dish where we would give them some sort of treatment, either in the incubator at a high temperature to see if the enzyme would degrade, or at room temperature to see if they were at high activity. So in essence, we were trying to figure out which mutants would be better than the regular or wild type. We would then isolate them in these boxes that we see on the left, where we would grow them and transfer them to these clear containers on the right, where we could actually send them in for testing to figure out which mutants are actually making better enzymes than before, which we would then send for genomic sequencing and publish the results for the better sequences. Thank you for listening to this presentation.